So hi, today I will talk about the genomic basis of evolutionary differentiation among honeybees. So what are the main characteristics that tells honeybees from other bees, apart from other bees? So honeybees build vertical comb, progressively feed their brood, and are extremely polyandrous, and have evolved symbolic dense language. They all, even if other bees also demonstrate nest thermoregulation, honeybees are highly precise to thermoregulate the nest. So they have also different further specificities linked with genome features. As you may know, usual hymenopterans are known to have a high recombination rate compared to solitary ones, but honeybees have extreme recombination rate and the highest recombination rate among all metamozoans. You may also know that hymenopterans compared to other insects have transposon depleted genomes, but again, honeybees have extremely transposon depleted genomes even compared to other hymenopterans. And finally, they have extremely AT-biased genomes. So when we think about honeybees, we think about the Western honeybees, Apis mellifera. But there is other species. Most of them come from Asia, and they can be divided into three groups: the dwarf honeybees, the giant honeybees, and the cavity nesting honeybees. So the three groups of honeybees have differences, and as the name says, they differ in their size, with the dwarf honeybees being small and the giant honeybees being big. And they don't differ only on their individual size, but also on the colony size, with again, the dwarf honeybees having the smallest colonies and giant honeybees having the biggest colonies, and the cavity nesting honeybees being in between. They also differ in their polyandry level, with dwarf honeybees having the lowest number of mating and the giant honeybees having the highest number of mating. And again, the cavity nesting honeybees are in between. As the name indicates, cavity nesting honeybees nest within cavities, while the two other groups are open nesting. They also differ in their nest thermoregulation and their abilities to regulate the temperature of the nest, with the cavity nesting honeybees being the highly precise, while the giant honeybees are not that good at it. And you can see, interestingly, that the dwarf honeybees, even if they are open nesting, are able to manage to keep nest temperature really precisely, with only 1 to 2 degrees variation. And they also differ in the migration pattern, with giant honeybees only having long distance migration. So what we did the de novo assembly of Apis dorsata, and with this we take the genome of Apis florea and Apis mellifera, each species representing one group of honeybees, and we take as our outgroup bumblebees impatiens, because it's most closely related to honeybees, and all bumblebees are primitively eusocial, while all the honeybees are highly eusocial. And we reconstruct the phylogeny from that. And we detect the positive selection on genes, on single copy orthologs, on chemosensory genes. And from those results, we check which pathway are enriched or depleted of fastly evolving genes. And we also look at lineage-specific genes. So when we reconstruct the phylogeny, that it agrees with previous phylogeny, showing that Apis florea, the dwarf honeybees, diverged first from the rest, from the common ancestors. I oriented the phylogeny according to the colony size. You can see that on the top, Bombus impatiens has a small colony size below 1,000, and Apis dorsata has a biggest colony size up to 50,000 workers. Interestingly, we can see kind of negative correlation between the colony size and the GC content of their genomes. And here in green, you can see the orthologs among the 4 species, and in blue, you can see the lineage-specific genes in the honeybees, with the highest number of lineage-specific genes in the Apis florea. So when we look at the result from the perspective of increased adaptation to increase eusociality, we found in the Apis ancestors that the functional pathway growth factor activity is enriched of positively selected genes. And we know that this pathway is linked with gene regulation. On the contrary, we found that most of the transcription pathway are depleted of fastly evolving genes. So we know that the adaptation to eusociality is linked with the increased capacity of gene regulation. In previous papers, studies have demonstrated that adaptation of eusociality is linked with positive selection of transcription pathways. Here we find that transcription pathways are depleted of positively selected genes, which denotates the subsequent conservation of these transcription pathways most probably to not disrupt the recent and complex adaptation to eusociality. On the contrary, on the other hand, we find other pathways involving gene regulation to be enriched of fast evolving genes, which denotates the adaptation to increase social complexity in honeybees. So when we look back at the Apis ancestors, we find also two pathways to be fastly evolving: cellular response to X-ray, which is basically the mechanism to repair double strand breaks, which is known to be involved in recombination, which might explain the high recombination rates in honeybees. We also found piRNA metabolic pathway to be enriched of fast evolving genes, and we know that piRNAs are primarily used for silencing transposons, which also could explain the transposon depleted genomes in honeybees. So when we look further, we see also that mitochondrial translation pathways are fastly evolving in Apis ancestor. And here is not mitochondrial genes, is nuclear genes. And we know that nuclear genes involved in the mitochondrial translations have been selected to increase translation accuracy. And we see this pattern not only in Apis ancestor, but also in Apis dorsata and Apis mellifera. In addition, in Apis dorsata, we found a pathway to be fastly evolving linked with the maintenance of genome integrity. So with increase of colony size, we see increased number of pathways to maintain the genome and proteome integrity. And we know that increased colony size in eusocial hymenopterans lead to high mutational load due to the low effective population size and the continuous oogenesis. Like in vertebrate spermatogenesis, the continuous oogenesis leads that the later egg is produced, the more meiosis it underwent, and with each meiosis, there is accumulation of mutations, which could explain the positive selection to maintain genome and proteome integrity. But even if maintenance of the genome and proteome integrity is primordial, for example, to maintain well-adapted phenotypes, it hinders genetic diversity, which is essential for adaptive evolution, especially in eusocial insects, where it enhances immunity and division of labor. And we see with increased colony size, we have increased polyandry, along with recombination, which drive genetic diversity. So we see a trade-off between the maintenance of genomic integrity and while generating genetic diversity in the honeybee evolution. So yeah, you can see such a gradient with increase of colony size here. And our results suggest also a convergent adaptation to open nesting in Apis florea and Apis dorsata, with in Apis florea, functional categories to be enriched of lineage-specific genes linked with nest thermoregulation, and also lineage-specific genes linked with nest thermoregulation and color resistance. In Apis dorsata, we have the same lineage-specific genes involved in color resistance, and also pathways that are fastly evolving linked with nest defense, suggesting both independently adapt to open nesting. So what, from our result, what we can sum up for the most plausible evolutionary scenario of honeybees, that they are generated from small colonies from single mated queens on developing nest on the horizontal plane within cavities, and then the honeybees ancestors adapted to vertical comb within cavities, which lead to increased colony size, and this increased colony size led to increased mutational load. And this high mutational load lead to a positive selection to maintain genome and proteome integrity. While this is balanced by the increase of adaptation to increase polyandry and recombination to maintain generating genetic diversity. Then we see that both Apis dorsata and Apis florea adapted to open nesting, which highlights the need to revisit the theory behind the evolution of the dense communication in honeybees. And we see further on that the Apis dorsata increase the colony size, which lead to higher mutational load and again to positive selection to maintain genome integrity, while balancing genetic diversity by increased polyandry. And we see the same pattern in Apis mellifera to a lesser extent. And with that, I would like to thank all my co-authors, in particular Olaf Rippel, Chris Elsik, and Philip Brands, and of course you for your attention. Thanks.